Recombinant protein purification is a common method used by biochemists to isolate and purify a specific protein of interest. In the following video, you will learn the basic steps involved in recombinant protein purification and be provided with an overview of common purification methods used during the process. To begin, recombinant protein purification follows 6 basic steps. These include gene cloning, transformation or transfection, selection, expression, cell harvesting, and finally purification. After purification, the isolated protein can be further characterized and analyzed using a variety of different techniques that will not be covered in this video. Before you can begin the purification process, you first need to decide on your expression system. There are a few common expression systems that scientists use. These include bacterial cell expression, yeast cell expression, insect cell expression, and finally mammalian cell expression. In bacterial cell expression, scientists typically use E. coli cells for overexpression of recombinant proteins. E. coli are cheap, easy to use, and can express large amounts of recombinant proteins. However, some drawbacks to using bacterial cell expression are the lack of post-translational modifications compared to animal cells, challenges in expressing larger or more complex proteins, and the effects on protein folding that may cause the proteins to easily degrade. In yeast cell expression, scientists typically use Cerevisiae, also known as baker's yeast. Similar to bacterial cell expression, yeasts also offer a cheap and easy method to express recombinant proteins with the added benefit of yeast also being a type of eukaryotic cell. However, using yeast cells also comes with its own set of challenges. For instance, yeast cells are very challenging to lyse, making cell harvesting and isolating of proteins difficult. Yeast cells are also still very different from animal cells, meaning important protein modifications or accessory proteins might be missing. In insect cell expression, scientists typically use SF9 or Hive5 cells. Insect cells can be used to express larger, more complex proteins and are more closely related to animal cells in terms of protein modifications. However, unlike bacteria and yeast cells, insect cells are more expensive and challenging to maintain. Additionally, the cloning process to prep recombinant DNA is more complicated and time-consuming. Finally, in mammalian cell expression, scientists typically use HEC293 cells. Mammalian cells are the most similar to human cells and provide human-like post-translational modifications that may be important to a protein's function. However, similar to insect cells, mammalian cells are very expensive and high maintenance, often requiring specialized CO2 incubators for proper expression. While each of these expression systems have their pros and cons, for the remainder of this video, we'll be focusing on bacterial cell expression as it is likely the first expression system you will encounter in the lab. Starting with gene cloning, we need to create the recombinant DNA that will be used to express our protein of interest in our host organism. Recombinant DNA is essentially artificial DNA created by joining DNA fragments from different sources. During the gene cloning process, small circular DNA molecules called plasmids are removed from bacterial cells. These plasmids serve as vectors that will be used to carry genes of interest that encode our target protein and match the expression system we plan to use. The gene of interest can be isolated using various techniques, but the most common one is polymerase chain reaction or PCR. This video will not cover the steps involved in PCR, but understand that this technique is used to amplify specific DNA sequences for further experimental uses. After isolating our gene of interest and identifying the vector plasmid we plan to use, the two DNA fragments can be combined using restriction enzymes to cut open the vector and ligation to paste in the gene of interest. The result is a new recombinant DNA. Once the recombinant DNA is successfully cloned, we introduce this foreign DNA into our bacterial cells through a process called transformation. Transformation is typically used for bacterial cells and involves uptake of DNA from the environment. While transfection is used for eukaryotic cells, and usually involves other methods of DNA delivery. After the bacterial cells have been introduced to the recombinant DNA, we select the bacteria that successfully took up the plasmid. This is done by adding antibiotics to the bacterial culture. Because the plasmid we introduce contains the genes for antibiotic resistance, only bacteria that took up the plasmid will survive. This leaves us with an enriched bacterial culture that is primed to express our target protein. Next, the protein is expressed by the bacterial host. This is done through typical transcription and translation of the gene of interest. To optimize protein expression and improve yield, scientists often include an inducible gene within the plasmid that controls when the protein is expressed. As a result, only during induction, when a specific inducer molecule is added to the culture, will a bacteria start expressing the target protein. Once the bacteria has been given enough time to express our protein, the cells are pelleted and stored for later use. When we are ready to start the purification process to isolate out our target protein, cells are harvested and lysed open. Cell lysis is the process of breaking open cells, causing the destruction of the cell membrane and the release of intracellular contents. Now that the cell is lysed, purification first begins by separating out the soluble and insoluble cellular components. Centrifugation is often used to separate these components based on size and density, with soluble proteins typically remaining in the supernatant, while the insoluble components, like cell debris and other proteins, form a pellet at the bottom. After this step, the supernatant is kept for further purification while the insoluble pellet is discarded. The remainder of this video will focus on the three main chromatography methods used to isolate and purify proteins of interest. Chromatography is basically a fancy term used to describe techniques that separate the components of a mixture. The chromatography methods this video focuses on include size exclusion chromatography, also known as gel filtration chromatography, affinity tag chromatography, and finally ion exchange chromatography. First, in size exclusion chromatography, protein mixtures are separated based on size. As a protein travels through the column, larger proteins elute off the column first. The elution of proteins can be monitored by reading the chromatogram, which is a visual representation of the separation of components that plots the detector's response, or signal intensity, against time or elution volume, showing peaks that correspond to different components of the mixture. But how does this column know which proteins are larger or smaller? Well, inside the column is a matrix of porous spherical particles that affect the movement of proteins through the column. Taking a closer look, smaller proteins get trapped in the porous network of the beads, while larger proteins either pass right around the beads or move through larger openings. This ultimately allows large proteins to move through the column faster and elute off sooner, while smaller proteins are slowed down and elute off later, creating separation between components in the mixture. Next, in affinity chromatography, protein mixtures are separated using an affinity tag attached to our protein of interest. During the cloning process, an affinity tag gene can be added to the recombinant DNA and fused to the protein of interest. When this protein mixture is then added to the affinity tag column, proteins without the tag will be washed away, while proteins with the tag will remain on the column until eluted off using a specialized buffer or solution. For histidine or his-tagged proteins, they can be eluted off the column using a high imidazole buffer, which disrupts the interactions between the his tag on the protein and the nickel affinity beads in the column. There are also other affinity tag methods such as FLAG, STREP, MBP, or GST, all of which have their own unique elution methods. Finally, in ion exchange chromatography, protein mixtures are separated based on charge. Since proteins are simply a string of amino acids, we can alter the overall charge of the protein by changing the pH of the buffer or solution it is in. For instance, when the isoelectric point, or pI, of a protein is equal to the pH of the solution, the net charge of the protein is 0. However, if we increase the pH of the solution so that the pH is greater than the pI, the protein has a net negative charge. In contrast, if we lower the pH so that the pH is less than the pI, the protein has a net positive charge. Similar to how proteins can have different sizes, proteins can also have different isoelectric points. This means while one protein might have a net negative charge at pH 9, another might have a net positive charge at the same pH. This difference in charge works perfectly for anion exchange chromatography that utilizes positively charged beads to trap negatively charged proteins. Proteins are then eluted off the column using a salt solution gradient that gradually increases in salt concentration and disrupts ionic interactions between charged molecules. As a result, the more negatively charged the protein, the tighter it binds to the beads, allowing for the separation of protein mixtures. Finally, after successfully purifying our protein of interest, we can use methods such as SDS page analysis or Western blotting to characterize the overall homogeneity of our sample and confirm our protein of interest is present. From there, structural biologists can take these protein samples and begin their structural and functional studies.